Oh, yes. Thank you for the invitation to talk at this seminar series. I have been working quite some time on the bio-based ground improvement. I'm reaching the almost 20 years right now. So, and I'd like to, well, show a little bit of an overview of what we have done in these last 20 years. So, to start with what is bio-based ground improvement, I'd like to refer to an excellent paper by Jason de Jong, one of the colleagues who's also has been very active in this field. And in general, it's like we're trying to use biology to stimulate a chemical reaction that may result in products like minerals, biomass, or biogas, and these products then affect the mechanical properties of the soil, which then eventually, if you apply it at a large scale, can be used as a ground improvement method. But then the main question obviously is how to do that, how to design a treatment procedure for such a bio-based ground improvement method. It starts by selecting a biological process, then inject the biological agents or substrates that the agents consume into the ground, wait until the reaction happens, and then if necessary, you may want to remove some unwanted byproducts. Sounds quite simple, but it starts with selecting the right process, and the soil is not the most favorable environment for all species. There's typically no light, limited oxygen, so any oxygen using bacteria or any phototrophic bacteria, they're not so easily survive in the underground. And then also, depending on what type of soil you have, you may not be able to inject bacteria or microorganisms in the ground. Particularly if you have clay soils, you cannot inject it in clay soils. So if you want to use biology, you might have to do some mixing. So the process I started with, and I think most people have been working on, is the so-called microbially induced carbon precipitation, often based on the reaction of urea hydrolysis, in which urea reacts with calcium chloride, in most cases calcium chloride, by using the enzyme urease, which hydrolyzes the urea into ammonium chloride and calcium carbonate. And then calcium carbonate minerals will strengthen the ground. So question now is, so this is a reaction, how to design a procedure if you know the reaction. So obviously you have to come up with some input variables, like you want to have a flow rate, you have to decide on what concentrations to use, how long to pump the nutrients into the ground, and maybe instead of injecting, you want to percolate it vertically, or you may want to mix it in. And you can choose different strategies to get those nutrients into the ground, and eventually, you want to predict, given a certain procedure, what the outcome will be. So the strength, the stiffness, variability, porosity, or cost. So the question then is, the procedure which you want to design is something like a black box, and you have to fill in the black box. So one way to do that is going into the theory, and the basic theory which is involved is reactive transport modeling, where we have a flow equation on the left side and a transport equation which shows the transport of nutrients. The change in concentration of certain species in the ground is a function of the dispersion, convection, and the reaction term. So first thing I started with when I started to investigate this problem was to investigate the reaction term. So when I look back at this reaction, it looks pretty simple, but I already split the reaction. So in fact, it's two reactions. First, the hydrolysis of urea. It's urea reacting with water and forming ammonium and bicarbonate. And while doing so, it consumes acid. So as a result of this reaction, the pH would go up. But if you do this in a water environment, there's a lot of other species which are in equilibrium with these reactive species. So if you want to properly simulate concentrations of each of these species, you have to take into account all kinds of speciation equilibria. maybe some gas equilibria, and maybe some complexation equilibria if you also add calcium to the system. So then the other half of the reaction is if you have calcium in the system and you produce bicarbonate, you get precipitation of calcium carbonate crystals, a solid phase, and then this precipitation, if you use bicarbonate as the main precipitating component, produces acid again. So altogether, the reaction should be pH neutral, and the pH should not increase as a result of that. But again, this is not so simple because there's calcium carbonate is a polymorph, which means that there's multiple different crystals. So you might form amorphous calcium carbonate, you might form vaterite, or you might form calcite. Each of these components can be formed. And if you want to predict what happens, you really need to include the different types of minerals. So what we do see is that the rate of hydrolysis and the rate of precipitation really affects the type of crystal. So here you can see at a very high bacteria concentration or a high reaction rate, we form so-called vaterite, spherical vaterite crystals. If we use a low bacterial concentration, we form so-called calcite crystals. Also, depending on the sand type, we may also expect different types of crystals. So on the left side, you see a quartz sand with a high reaction rate, and you see that again the spherical vaterite type of crystals between the sand grains. While on the right side, you see a carbonate sand at the same reaction rate, but in this case, the carbonate particles themselves act as a nucleus for the crystallization process, and they get covered by very many tiny calcite crystals as a coating on top of the existing sand grains. So same reaction rate, but different sand type gives completely different precipitation characteristics. So that's the type of reactions you take into account, and then you have to look into how fast does the reaction go. So what components affect this reaction rate? It's the amount of urease. The more urease you add, the higher the reaction rate. It's the type of urease. So many of people doing research in this field have been using Sporosarsina pasteuriae, and they found it is one of the species of bacteria which has a very high urease activity. If you look all the way down, you see jackbean. This is a plant species which is also rich in the enzyme urease, and it's the one which is typically commercially available in the lab. And nowadays there's also methods developed to extract them from the raw beans. And both methods are quite competitive in terms of cost of enzyme. Then, depending on the amount of substrates, typically if you have urea in the system and you add urease, at high urea concentrations, there's sufficient substrate available, so the reaction reaches a maximum rate. But if urea starts to deplete, then the concentration of urea starts to go down. Also, the reaction rate goes down. Then, if you have calcium in the system, if you add too much calcium, the reaction rate may also go down, because typically bacteria, but also the enzymes, they do not like very high salt concentrations. And then, depending on the pH, well, luckily the process is a bit pH neutral. So typically the process happens at the maximum rate. But if you would operate under very acidic or very alkaline conditions, also those conditions are not favorable for the hydrolysis process. Then temperature, if you increase temperature, the reaction goes faster, like many biological processes. And there's additional factors like the amount of calcium carbonate formed. Eventually, bacteria get encapsulated and they do not participate in the reaction anymore. And also, if you don't feed the bacteria, they just decay in time. So overall, if you add up all these processes, you see that the reaction depends on many different factors. Question is, which process do you need to take into account? This is just the first reaction of hydrolysis. If we also look at the precipitation reaction, we see that we can describe the precipitation reaction as a function of the supersaturation S. If the supersaturation, it means the square root of the ionic activity product over the solubility product. If that becomes larger than 1, then precipitation rate may start, given a certain rate constant Kp. This rate constant is in fact not a constant. It's a product of the amount of surface area, the amount of nuclei in time, divided by the molar volume times the actual growth rate constant. So if you're in a system where the surface is constant, then it may actually be a constant parameter. But typically, if you have growing crystals, then the surface area of the crystals available becomes larger and larger. So eventually, the Kp constant actually becomes larger as well. So then your precipitation rate varies depending on the actual bulk kinetic constant. So, well, if you also want to include the nucleation kinetics, so before a crystal can start growing, it has to first nucleate in a solution. So if there is no nuclei present, you have to exceed a certain barrier, an energy barrier, before actually crystals can start growing. So you first have to form nuclei before you can start growing them. Unless there's already nuclei present, then, or existing surface present, then you may find that you get heterogeneous nucleation. Anyway, quite complex nucleation theory. You may want to include it if you want to simulate it appropriately. So what I've done in my PhD thesis at the time, I tried to find out what level of complexity is now necessary to actually simulate or predict what happens in this process. So I started with the single reaction which we started with. Then I included all the equilibrium reactions, and eventually I also included precipitation kinetics, and I looked at the differences between the different models. So first, starting with a single reaction, which is this 1 urea plus 1 calcium chloride goes to 1 calcium carbonate. And you can see that as you consume urea, you produce calcium carbonate and you produce ammonium. So quite a simple model. And I also assume here that the hydrolysis rate is equal to the precipitation rate. So if I include all the equilibrium reactions, I still see that the dominant concentrations are still urea and calcium, and they're almost equal. At the same time, calcium carbonate increases and ammonium increases twice as much. But because I include all the equilibrium species, I can also predict the pH. And what I mentioned in the beginning is that if you start a reaction, initially pH rises, but then it drops again, and it remains around neutral between a pH of 6.5 and 7.5 during the entire hydrolysis process, depending on how much urea and calcium you add in the system. So then, well, I had the predictive model, but then I looked at the actual measurements, and I saw that the measurements are really not fitting the simulations in terms of pH, in terms of salinity, which is this K norm or normalized electrical conductivity. You see, I'm almost there. During the measurements, I slightly increase the electrical conductivity a little bit faster than in theory. But in terms of pH, I'm almost one pH, one order of magnitude different, or I'm almost one order of magnitude off from the actual proton concentration in the solution. You may argue, wow, this is not fitting at all. On the other hand, if you consider that the pH means the minus logarithm of the proton concentration, I can still predict the pH with, well, 10 to the minus 6 accurate, starting from a 1 molar solution. And so it's 1 micromole of protons, or a tenth of a micromole of protons. That's the difference between pH 7.5 and pH 6.5. So it's actually quite accurate, although the pH is very sensitive depending on what process you include. If you now zoom in at the beginning, it was actually quite interesting to see that during the experiment, pH rises initially and then drops down, rises again, drops down, rises again. It forms this pattern. And what we, well, at that moment thought, okay, what's happening here? It seems that well, you could explain this type of pH behavior by multiple nucleation events. So initially you start building up carbonates as you hydrolyze urea until you exceed this energy barrier where nucleation can, oh, where nucleation can start to occur. And at that moment, you get a sudden nucleation event, but as it nucleates, the pH drops again, so your equilibrium goes down, and gradually pH can go up again. So, well, this is a hypothesis, but I have not been able to really predict this behavior appropriately. So at that moment, we said, okay, bulk chemistry, we can actually well predict with just a simplified single reaction model, as the precipitation rate is almost equal to a hydrolysis rate in terms of bulk kinetics. However, if you really want to predict the strength, and we've also found out, or other research have found out in looking into mechanics, that the strength really depends on the type and the number and the size distribution of the crystals. You have to include the precipitation kinetics, and so far I haven't seen anyone to be able to predict the actual precipitation kinetics appropriately in such a heterogeneous system as a soil. What we do see, and there are some trends. So if you do a very fast hydrolysis reaction, typically get higher degrees of supersaturation, which means there will be more crystals and smaller crystals, and also probably more metastable mineral phases like amorphous calcium carbonate or craterite. That's a general observation we see, but still we find that to really quantitatively predict the type and the size and the mineral of the calcium carbon minerals is quite difficult because the soil is just so complex that it's very difficult to really predict that. So I would argue it's still ongoing work in theory for many years and many researchers, but it will be impossible because in the end, we cannot define all the input parameters in an open soil system. We can maybe predict them, but then in the end, you still want to develop a treatment procedure, or you want to predict what will happen if you, whatever you pump in the ground. So if you want to make prediction, the challenge is to select and validate the proper assumptions. So when am I allowed to use a simplified reaction equation, and when am I not allowed to do that? So that's the basics of the theory. But yeah, we don't have an infinite number of years to study. So the other approach towards trying to get this done is by doing experiments. And also during my PhD, I was very lucky to work with the contractor and the research institute. And we were able in a relatively short time span to start with a small column done by Vicky Whiffen, one of the other pioneers in this field. She demonstrated that she could cement the sand column. And then the company and the research institute I was working for, they said, well, one of the potential advantages of this ground improvement method is that we can actually treat at a long distance. So can we try to get a long distance, because most of the other ground improvement methods are not able to do that. So within a year or two, we managed to get the right concentrations of bacteria and the right nutrients, that within a few flushes, we were able to treat it over 5 meters length. And a bit later, we were also able to get it relatively homogeneously. So the next stage was, if you do a full reactive transport or a full injection method, then real injection is not 1D, it's actually 3D. So we started with 1 cubic meter, and once we've done this together with the contractor who was involved, they said, okay, we can do it better. And then we developed this 100 cubic meter sandbox experiment, where on the left we had 3 injection wells, and on the right 3 extraction wells, and a lot of monitoring wells in between. So based on this progressive insight during the experimental campaign, we were able to get a successful experiment done in which we gave it 5 flushes of 1 molar calcium and 1 molar urea, preceded by about 1 pore volume of bacteria suspension. And after these 5 flushes, we excavated the removed loose sand, and we found that about 40 cubic meters was solidified. But within the solidified mass, there was quite some heterogeneity, with calcium carbonate contents ranging between 0 and 25%. So anyway, so that was the conclusion after these large-scale experiments. And after having done that, we start to think about, okay, what can we do with this? And at that time, we were not the only ones. So also at Montana State University, they are doing a lot of work on this process. And they came up with a nice review paper showing all potential applications. What can you do with this urolytic biomineralization? And at that time, because we were working with a contractor, they said, okay, we might have a potential application, which is stabilizing gravel when we start drilling for horizontal directional drilling pipeline through a gravel layer, because they said, well, it may potentially collapse when we start pulling this pipeline through the gravel. So in order to prevent that borehole collapse, they said, well, what if we stabilize the gravel layer? At that moment, we had to redo our preliminary experiments and scale up procedure for gravel. But once we showed that we can actually cement gravel and drill a hole through and leave it open, the client and contractor were sufficiently convinced that this method may work as a risk mitigating measure. So this is what it looked like with the injection wells and extraction wells. On the right upper side, you see the tanks with the urea and the calcium chloride, and then in the middle, the mixing tank, and then the manifolds for the flow lines going towards the injection wells. So we injected a total of 4 by 4 by 50 cubic meters of sand and gravel, ranging of a depth between 4 and 20 meters. And installed injection wells in the 5-meter grid. So then we did some monitoring, we did some geophysics, electrical conductivity or resistivity, georesistivity measurements showing elevated salt concentrations where we had injected, and we were able to excavate some samples and showing some stability. However, results were not conclusive in the sense that we could not really identify increase in strength in the gravel layer because sampling or testing gravel in the field proved to be quite difficult. However, the contractor was happy because they were able to install the pipeline without any problems. So after that, many other people have started on pilot studies, and the last one I had been involved with is shown here on the right side, where we did some soil stabilization to create this landscape art monument, excavated in a loose sand, and they wanted to allow these trenches in the shape of a peace symbol to stay upright. Therefore, they wanted to cement the sand. It seemed to be quite a good pilot because, as I mentioned earlier, the cementation is relatively heterogeneous. So therefore, you don't want to yet test it in an actual construction site where there's a lot of loads involved. But in this case, the trenches were allowed to collapse slightly, but they just wanted to increase the longevity of those trench slopes, and therefore this process seemed to be appropriate. So after having done those field trials and also looking at all the other field trials that have been done since, we can say that MICP biourea hydrolysis can be used to stabilize soils. There's many different studies on various injection, mixing, and monitoring and sampling methods available. Empirical correlations have been developed for strength, for stiffness, for liquefaction resistance, and there might be many possible applications for it. However, based on the trials in 2010, we found still limited evidence in the field, limited control of the homogeneity of the cementation process, and there were significant costs involved, up to about 400 euros per cubic meter of soil, which is similar to, let's say, jet grouting. So from an economical perspective, it was not yet competitive compared to traditional ground improvement methods. So you really have to look into an application where there's no alternative application possible. Well, also the environmental impact. We started this research by thinking, okay, if we use biological method, maybe more sustainable. But if you see the type of equipment you used and all the resources, it's still consuming quite a lot of resource. You also produce the ammonium chloride, which is the byproduct which has to be removed. And you have to cultivate bacteria. So all these processes and products that require energy and resources. So you really have to do a life cycle sustainability assessment in order to evaluate the environmental impact. So what we did do next, and we tried to optimize the process. We looked at alternative applications for which we might not need as much substrates. We also looked at alternative processes, and finally, we also looked at alternative products. I see a hand raised. Do we do questions in between?
Yeah. No, don't worry. I mean, we'll take all the questions at the end.
Okay, that sounds good. So first thing, look at alternative applications. Once I graduated from my PhD, I could start as a lecturer at TU Delft. And we got a research program funded looking in alternative applications. And the most promising applications we looked into were beach stabilization and liquefaction mitigation. Why? Because both applications require actually a very low strength increment and also very limited homogeneity. So it may not be necessary to create a large strength concrete block, but just a little bit cementation would be sufficient to mitigate liquefaction and also to increase the stability of beaches. And the pictures you actually see here are not from MRCP, but actually from another biological stabilization process using sand worms or honeycomb worms, which actually cement the sand not by calcium carbonate, but by using some organic polymer. So also it will be an alternative product to look into, which has not been significantly investigated yet. So, but what we also looked into was alternative processes. As alternative process was denitrification. In this case, we would use calcium fatty acids and calcium nitrate as resources that would cement the sand by producing calcium carbonate. But this process would not require special enzymes and special microbes to add, but we could actually grow these microbes using the substrates as they would feed on nitrate and fatty acids and produce calcium carbonate as a byproduct together with nitrogen gas. So also we would not have to remove the byproduct ammonium chloride. So looking into that process as an alternative process to produce calcium carbonate, again, from the theory, we could predict the different kind of reactions, what chemical reactions are happening. And here, the chemical reaction would depend on the amount of biomass growth. If the biomass would grow very fast under favorable condition, then a lot of the carbon used, a lot of the acetate would go to biomass, where if we would, if the bacteria would grow slower, relatively we would produce more calcium carbonate. But also we would produce nitrogen gas and a little bit of CO2, which probably would generate gas. And well, based on the stoichiometry and the ideal gas law, we could predict how much gas would be formed and what the residual saturation would then be in that case. So as you can see at very low concentrations, for example, 20 millimolars of substrates, nitrate, you would already desaturate the soil in its very shallow conditions, let's say 5 meters below surface, up to 20%. And if you desaturate the soil, that would probably also fill up the pores and might affect the distribution of your substrates and products. So, but well, we had to use relatively low concentrations in denitrification. Why? Because if you use too high concentrations, some of the nitrate may not be converted because the bacteria, denitrifying bacteria, they do not like too high nitrate concentrations. So we had to keep the concentrations low, which meant that we had to apply many different flushes, or well, up to 3 to 15 flushes to reach about 1% calcium carbonate. And if you compare it with the urea hydrolysis, there we could get up to 25% calcium carbonate within 5 flushes. So from that perspective, the urea hydrolysis is a bit more efficient in time and number of flushes. Also, reaction rate is a little bit slower. So to get this 1% required 1 month, whereas in the previous example, to get 10 to 20% calcium carbonate, you would only require 2 weeks or so. But still, we were able to cement it, and we could demonstrate a little bit increase in stiffness and a little bit increase in differential stress at small strains. So, for example, for liquefaction mitigation, this might just be sufficient. And the great advantage is that you don't produce the ammonium chloride, so you don't have to think about any remediation process to get it out of the ground again. So one of the well, I mentioned, one of the byproducts was the nitrogen gas and the CO2. But we expected if you have many different flushes, you produce more gas, but at a certain moment, you create a continuous gas phase and the gas may vent out of the system. And doing sand columns experiment, we found that actually its saturation, ranged between 75% during production of the nitrogen gas to 90% during the next flush when we flushed in new substrates. However, even though the degree of saturation remained almost constant, still each flush generated more biomass and more calcium carbonate. And after several flushes, we actually found that the permeability did decrease significantly, even though there was a relatively small amount of porosity reduction by the generation of biomass and calcium carbonate. We still found significant reduction in hydraulic conductivity. And one of the explanations for that, we think, is that the gas bubbles sit in the middle of the pore and it forces biomass and calcium carbonate to form at the pore throats, thereby very efficiently clogging the system. So even though we could cement the calcium carbonate, there were a few disadvantages. So we said, well, why not look at another process or another product? And actually the same reaction also generated the nitrogen gas. So if we would introduce nitrogen gas in the ground, we could actually use that to mitigate liquefaction. Here you see first the fully saturated soil, a loosely packed, fully saturated soil. If you start to shake it, you get contraction, so you get pore pressure buildup, thereby liquefaction. The idea was if we now partially saturate the soil, the shaking may cause it to contract, but because the gas is compressible, it actually dampens buildup of pore pressure and thereby mitigating liquefaction like an airbag in the car. You're dampening the impact. Colleagues here in Singapore, Professor Yang Chu and his student, and now also professor in China, Jia He, they did a lot of testing in this field, and they demonstrated with a relatively large-scale experiment that you only need up to 20% degree of saturation to significantly dampen these pore pressures during cyclic shaking events. So if we now compare precipitation process with the desaturation process, it actually shows that in order to produce 10 to 20% nitrogen, you only need a single flush with less than a kilogram of substrate per cubic meter. If you compare that with precipitation, to get 1 to 3% calcium carbonate cementation, you require up to 25 to 70 kilograms of substrate per cubic meter, and you require 3 to 10 flushes to bring it in. So desaturation to the target level only would require 40 times less substrate. So that was promising when we found that. So then we said, okay, it may be promising, but now we have to think, how is this gas distributed when we generate it? And will it be persistent? So here we can dive again into the theory. It requires, thinking about the reaction, thinking about the transport, thinking also about the transport after we have treated the material. And several theoretical models have already been developed, which are able to predict what happens with the gas once it's trapped in the ground. So here, in an illustration, what desaturation would do with the monotonic shear strength. You can actually see that the black line here is an untreated monotonic triaxial test at 100 kPa shear strength, and if we desaturate it by 6.25% gas, 12.5% gas, and 25% gas saturation, you see actually that the excess pore pressure goes down during monotonic shear of a loose black sand, while the Davy torque stress actually goes up. So we've done lab tests, and then the next question was, can we also demonstrate this in the field? At that moment, we found some colleagues in Portland State University. They managed to get a grant, and then we collaborated with them to actually try to find a demonstration site for this desaturation, for this microbially induced desaturation. So here you see a map of the city of Portland, and all the purple area is actually very highly susceptible to liquefaction during a very large earthquake. On the left, the Harborton area, this is a critical energy hub for the city of Portland. So there's a lot of oil and gas storage tanks situated down there. And on the right, the Sunderland side is very close to the international airport in Portland. So both sides are very critical infrastructure. So it would be very large catastrophe if these infrastructure sites would be affected by liquefaction during such large earthquakes, which are expected in the not too long future. So here you can see a little bit an image about that field trial is important. So you can see the geometry with the single injection well in the middle, several extraction wells on the outside. And we started pumping in nutrients for about 20 days. And well, we can see on the left, the injected blue line is the concentrations which we injected, and the red line is what we extracted, and the difference would then be what has been converted during treatment. So during treatment, we monitored the degree of saturation using in situ TIRS 12 sensors, and they demonstrated actually that the degree of saturation would drop to about 92%. And even after the treatment of, well, in this case, yeah, it's 30 days actually of treatment, and we saw that it continued to stay low. And then we continued to monitor. Here, these graphs, they show 120 days, but we have been continued to monitor it since 2019. And over the last 5 years, particularly the deeper sensors, both in pressure wave velocity and in the TIRS 12 sensor, both demonstrated continued desaturation for periods of almost 5 years now. So at the end of treatment, we actually asked Ken Stokey from University of Texas to come to Portland and try to liquefy the soil using their big shaking machines. But they were not able to sufficiently excite the soil in order to demonstrate that it would liquefy. So you could argue it has been successful, but also on the non-treated sites, comparing the non-treated side with the treated sites, that didn't show that much difference in pore pressure buildup. So we need to find a way to get bigger shakes in order to retest whether this method has worked. So microbially induced desaturation, based on these field trials, we can conclude that it can be used as a bio-based garden improvement method. Again, we continue to develop new injection mixing and monitoring sampling strategies, and we were able to reduce the cost by just focusing on the degree of saturation reduction. But still, similar to the MICP, we still have limited evidence for liquefaction resistance at field scale, and we're still working on that, trying to find new sites to treat it at large scale. Quality assessment, quality control methods. We don't yet know how to assess the quality. Yeah, pressure wave velocity is one way, but they can only test relatively local where the gas is situated, and doesn't really show at a small scale where it is. So also it will not show how the gas is distributed at the pore scale. So we still may need to develop new theory to predict how the gas would be distributed in different soil types, particularly because soil is relatively heterogeneous. It will probably sit in the larger pores, but then you have to know how the soils are stratified, how they are distributed in order to make a prediction of how the gas is distributed. And once you know how it's distributed, you still have to find out how it would behave under an earthquake. So some work we have been doing recently, looking into the effect of stratification in soils. And here you can see a video of an injected nutrient solution, which gradually gets consumed. And then because the red color disappears, you see that it actually generates gas, which then pushes the liquid to the inside of the tank. Also, you can see that even if you use a relatively low density fluid, that there's some fingers going down from the coarse layers into the finer grained layers, which affects distribution of substrates and thereby also the distribution of the gas. More recently, we've done additional tank experiments. Now we have a sand and a silt layer. Again, we injected the material and we try to monitor how the substrates were consumed. And I think, considering the time, I will proceed a bit faster. So here you can see again a second injection, and what was interesting now, you can already see that the injection pattern is slightly different because of the presence of the gas. But as we were producing the gas, also the gas was pushing liquid through the upper silt layer here, the white layer, and you can see some silt being eroded, forming these small erosion channels, erosion or steepage-induced erosion volcanoes on top of the silt layer, generating gas leakage paths. And eventually, if we continue to produce gas, we saw that at some point the gas was actually reaching the upper silt layer, the white layer, which was then just lifted up by the trapped gas. So you can also see that you need some confining pressure in order to maintain this gas in the sand layers. All right. So to conclude, some future opportunities, I think, well, we have demonstrated by now that bio-based ground improvements methods exist and that they do work. However, I haven't seen so many, although there's a few initiatives now. Commercialization of these technologies. Why? Because you have to come up with a competitive advantage compared to the existing methods. Therefore, you have to find the right application, get industry involved, and do some field scale validation. In many cases, they say, well, it's interesting, but do you have evidence that it actually works? And so far, the evidence is often not yet enough. So you have to do good marketing in order to convince a client to accept such a method, even though it has not been demonstrated that much. So I now only highlighted shortly two processes. There's many more different processes which we can investigate, and also many applications to explore. And so nowadays the field has been established. I'd just like to highlight there's an actual journal on biogeotechnics which well, every 2 months or so there's a new issue showing a lot of new papers in this field. And I would like to advertise a conference we're organizing next year on this field at Arizona State University. So right now I'm working for the contractor, and I found that for the applications Boskalis is interested in, which is mostly large-scale reclamations, the process is actually quite expensive. And the process of denitrification and precipitation, so there it has competitive advantage for places where there is already existing infrastructure, but if you have a plain field, then still dynamic compaction or rapid impact compaction are often economically preferred over any types of cementation or in situ treatment method. So therefore, I looked into, can we now maybe improve the efficiency of the dynamic compaction using desaturation? Similarly to a Proctor test, if you desaturate the soil, you would expect, because the gas is compressible, that you can actually better compact the material. I'm also looking currently in the use of worms or plants to accelerate consolidation of soft sediments. And within our company at the environmental department, we're also looking into using worms, shrimps, mangroves, and corals for ecological restoration and coastal protection. And so there's still a lot of applications to explore and new biological processes to investigate whether they would work or not work. And finally, what also interests me a lot is to think about climate change. Obviously, we all try to go back to carbon zero, but well, the main thing here is that we're aiming to try to go to carbon net zero. And for that, I would argue you have to look into solutions that nature can provide. So if you look at the total carbon emissions from the anthropogenic influence. Obviously, this is relatively large. This is already an old picture. 5.5 gigatons of carbon were produced. But if you look at the natural cycles, there in the ocean, it's about 90 gigaton of carbon per year, and on land, it's actually about 120 gigatons. So, just by changing the natural cycle by a relatively small percentage, can actually compensate for a lot of the anthropogenic emissions. So one of my missions is to also look into those natural processes of carbon sequestration. And with that, I'd like to thank all the people I've been working with in the last 20 years or so. And then I'm open for any questions.
Yeah, thank you so much, Leon.